Hello everyone, I am Mohamed Shafi, a postdoctoral fellow at National Center for Biological Sciences from India. I am going to talk about the work on structural analysis of the sodium transporter HKT1 in Oryza species for new determinants of salinity tolerance. Salinity stress is a major concern to the plants worldwide. Cultivated rice Oryza sativa is highly sensitive to salinity stress. Sodium accumulations in the shoots are one of the main reasons of this salinity stress. High affinity potassium transporters (HKTs) are plasma membrane transporters that release sodium from root xylem vessels and thus regulating or minimizing the sodium accumulations in the shoots. So far, HKTs are mainly studied in halophytic rice species Oryza coctata. It is an allotetraploid cultivated mostly in the coastal region of India and Bangladesh. Our study identified crucial residue variations near the pore entrance of HKTs that controls the ion transport. Now, if we look at the structure of HKTs, it contains four identical transmembrane pore lobe transmembrane domains. Each pore lobe contains selectivity filters, in which the first pore lobe selectivity filter determines whether sodium or sodium and potassium can be transported. Since serine instead of glycine is present in the first selectivity filter of rice HKT1 it transports sodium only. Furthermore, if we look closely to the secondary structure, in each domain we could see 5 to 6 transmembrane helices going from inside to outside of the membrane and coming back to inside. We could see a parallel helice is missing in the third domain, suggesting it need not to be identical also. Now coming to the modeling part, generating homology models for Oryza coctata HKT1 was the most challenging part in this study. No crystal structure has been solved for HKT1 so far, as this membrane protein is highly dynamic in nature. What we have is two crystal structure of potassium channel proteins from bacteria and fungi. Both of them have around 20% sequence identity to OCHKT1 protein, which is very less similarity. But if you look at the secondary structure of potassium channel and OCHKT protein, which we could see most of the helices are conserved. In these templates, we selected 4J7C as main template to model core region, as it is structurally highly similar compared to 3PJZ. And the missing part like N-terminal was taken from 3PJZ. We generated successful models after several loop refinement and by relaxing the structure by simulations. We also mapped the important residues controlling ion approaches that are known previously. The structure finally validated using Ramachandran plot and PROSA. Next, in our simulation and kinetic study, we observed HKT15K homologue from Oryza coctata what we have modeled, conferring a low affinity towards sodium ions. It was surprising to us that when we compared to cultivated rice OS HKT15 that showed higher affinity than a halophytic species like Oryza coctata. So we selected another homologue from Oryza coctata HKT15L, which is almost 80% identical to the K homologue and modeled using similar protocol. Then the residue differences near pore entrance was investigated between these two models. We could infer a swapping of positively and negatively charged amino acid residues near the pore entrance. The surface electrostatics of both model structures clearly showed the difference in charges of residues at the pore entrance. The positively charged lysine residue patch close to the pore entry site could repel the sodium ion and may play a more functional or regulating role in the entry. For HKT15L, this residue was replaced by a negatively charged glutamic acid might attract the sodium ion. In summary, comparing cultivated rice and Oryza coctata homologue showed variations in the residues near pore region. These opposite charges from OCHKT homologues also can form salt bridges and the pore region can contract. The positively charged residue at the pore entrance could be one of the reasons for the low affinity compared to cultivated rice OSHKT. Also, a negatively charged residue at pore entrance can give advantages in ion entry affinity for OCHKT-L over K homologue. Further, transport kinetics and molecular dynamics simulation studies are in progress. Thank you. I would like to acknowledge Professor R. Saudamini from NCBS, Dr. Gayatri from MSSRF, and Professor Anne from IPSIM. And this is a collaborative work between all these people. And also I would like to acknowledge my funding from Infosys Foundation, IPAP, and CEFIPRA. I also would like to extend my thanks to my colleagues from CAPS Lab.